Welcome to the Deep Dive. Today, we're tackling the very architecture of life human tissues. We're aiming for true foundational mastery.
That's right, we're going deep today. Think about it. How did we get from single, free-living cells floating around to these incredibly complex, coordinated communities? That's the journey we're exploring.
It really is mind-boggling. Over 200 distinct cell types in our bodies, but they all share the exact same DNA blueprint.
It all comes down to specialization, doesn't it? Different cells turning on different sets of genes. So today isn't just about naming things, it's about connecting the dots, structure, function, and crucially, the molecular basis that holds it all together.
And our mission here is to really master this. We're talking the 4 fundamental tissue types, the vital extracellular matrix, the scaffolding, plus specialized things like glands and membranes.
Absolutely. Think of this as the essential anatomical foundation. If you really grasp tissue architecture, understanding how every organ system works becomes, well, significantly easier. It's the bedrock.
Okay, let's rewind to the beginning then. Why bother forming communities? Why did evolution push cells together like this? It couldn't have just been about getting bigger, right?
No, not just size. The real advantage, the key selective pressure, was control.
Control.
Yeah, control over the internal environment. When cells started aggregating and specializing, working together, they could create and maintain a stable internal world, a sort of protected bubble.
Ah, okay. Like building a walled city instead of living out in the open.
Exactly. And that internal stability was absolutely necessary for really complex, specialized structures to evolve. Think about the brain that couldn't exist without a highly controlled environment.
So these cellular aggregates, these communities, that's what we call tissues. Groups of cells carrying out coordinated tasks.
Yeah.
Looking under the microscope, we've got a pretty reliable way to sort them out, right, based on structure.
We do. There's a really useful 4-pillar classification scheme. It covers almost everything. So first up, epithelia.
The linings and coverings.
Precisely. Continuous layers, cells packed super tight, very little space between them. They're the walls, the barriers, the surfaces.
Okay. Pillar number 2.
Connective tissue, or supporting tissue. Here, it's the opposite picture. The cells are often quite sparse.
Right, they're scattered.
They are. And they're embedded in this extensive extracellular matrix, or ECM. The ECM is really the star here. It provides the bulk, the strength, the shape.
Makes sense. Third.
Muscle. This one's straightforward, specialized for one main thing, contraction. Generating force, creating movement.
And the 4th pillar.
Nervous tissue, the communication network. Cells specialized for conducting and transmitting signals, electrical, chemical, super fast. Plus, importantly, all the supporting cells needed to keep that network running smoothly.
Okay, so that's the structural view. Epithelia, connective, muscle, nervous. But you mentioned earlier, this isn't just descriptive. There's molecular proof backing this up.
There is, and this is where it gets really elegant, moving us towards that mastery level. We can look inside the cells at their cytoskeleton, specifically at the intermediate filament proteins.
The internal scaffolding of the cell itself.
Exactly. And these proteins act like a molecular fingerprint for each tissue type. It's incredibly powerful, especially in diagnosing diseases like cancers.
How so? Give us the specifics.
Okay. For all epithelial tissues, the characteristic intermediate filaments are keratins. If you find keratins, you're looking at epithelium.
Right. Keratin's epithelium.
For connective tissue, the fingerprint is vimentins.
Vimentins collagens.
For muscle tissue, it's desmins.
Desmond's muscle.
And finally, for nervous tissue, you'll find neurofilaments and glial fibrillary acidic proteins, GFEs for short.
Okay, so let's test this. A pathologist gets a biopsy, maybe from a lung mass. They stain it, and it lights up positive for keratin. What does that tell them instantly?
Instantly. It tells them it's a carcinoma, meaning it originated from epithelial cells. Doesn't matter how weird it looks, or even if it's spread far from the original site, that keratin signature points straight back to its epithelial origin.
And if it's stained for vimentin instead?
Then it's likely a sarcoma, originating from connective tissue. Completely different type of cancer, different prognosis, different treatment plan. This molecular validation is why this 4-category system is so fundamental and, well, robust.
But biology loves exceptions, doesn't it? Are there cells that kind of blur the lines?
Oh, absolutely. Nature isn't always neat boxes. You definitely find cells with features of more than one type. We'll talk about myofibroblasts later. They look a bit like fibroblasts, which are connective tissue cells, but they can contract like muscle cells.
Interesting.
Or think about neuroepithelial sensory receptors. They're epithelial cells, but they're specialized to directly interact with the nervous system. Even the ependymal cells, lining the brain ventricles, they function like support cells for neurons, but they still hang on to their keratin filaments, revealing their epithelial roots.
So these exceptions don't invalidate the categories, but they maybe hint at shared developmental pathways.
Precisely. They show the evolutionary connections. But the four main pillars remain the absolute foundation for understanding tissue architecture.
Okay, foundation laid. Let's dive into the first pillar, epithelia. The body's boundary layers. You called them walls, gates, pavement.
Yeah, they have incredibly diverse jobs. Their primary role is often as selective barriers. They control what passes through. Sometimes they facilitate passage, like absorbing nutrients in the gut.
And sometimes they block things.
Exactly. Inhibiting passage, like preventing toxins from getting in or stopping dehydration. They also provide physical protection against scrapes, chemicals. And many epithelia synthesize and secrete substances or act as sensory surfaces, like taste buds.
And you mentioned that developmental link between epithelia and nervous tissue, both originating from the embryonic ectoderm. It makes sense seeing them interact so closely in the body.
It does. Structurally, the defining feature of epithelia is just how cellular they are. Cells jammed right up against each other. Very little extracellular stuff between them.
Except for that base layer, right?
Right. The only significant ECM they have is the basal lamina, which they sit on like a foundation. And the reason they can stick together so tightly and form effective barriers is down to intercellular junctions.
These molecular connections holding them together.
Exactly. And they're not just randomly placed. There's a specific sequence, a junctional complex, typically seen near the apical surface, the side facing the lumen or the outside world.
Okay, walk us through that sequence from top to bottom.
Starting at the very top, most apical junction is the tight junctional zone, also called zonulae occludantes. This is critical. It forms a seal, like a zipper, between adjacent cells.
And that seal forces things to go through the cell.
Precisely. It blocks the pathway between cells, what we call paracellular transport. This forces substances to take the transcellular route, passing through the cell membrane, into the cytoplasm, and out the other side. That's what makes the barrier truly selective. The cell gets to decide what passes.
Okay, tight junctions for the seal. What's next down?
Below the tight junctions, you typically find the adherent junctional zone. This provides mechanical strength, linking the cytoskeletons of adjacent cells, helping the whole sheet resist stretching and tearing.
Like a reinforcing belt.
Kind of. And then further down, you have discrete desmosome junctions. Think of these as molecular spot welds or rivets. They provide really powerful anchoring points, handling significant mechanical stress.
So tightly packed, strongly linked. But how do they eat if they're sealed off and don't have blood vessels?
That's a crucial point. Epithelia are fundamentally avascular. No capillaries run within the epithelial layer itself.
So no direct blood supply.
None. They rely entirely on diffusion. Nutrients and oxygen have to diffuse across the basal lamina from capillaries located in the underlying connective tissue.
Ah, okay. And that must limit how thick an epithelium can get, right?
Absolutely. Take a hard physical limit. If an epithelium gets too thick, the cells on the surface would be too far from the nutrient source. They couldn't survive. That's why living epithelia are relatively thin.
And because they're often on surfaces facing wear and tear skin, gut lining, they must need constant replacement.
They do. They generally have a very high regenerative capacity. Cells are constantly being lost from the surface due to abrasion or damage, and they're continually replaced by cell division, usually in the deeper layers near the basal lamina.
When we see epithelia combined with other tissues, there are common patterns, right? Like mucous membranes.
Yes. A mucous membrane, or mucosa, is a common structure. It consists of an epithelium plus its underlying supporting layer of loose connective tissue called the lamina propria, and typically it's kept moist, often by mucus glands. Think of the lining of your gut or respiratory tract.
And the other common one is the serous membrane.
Right. A serous membrane, or serosa, lines internal body cavities that don't open to the outside, like the chest cavity or the abdomen. It's typically a layer of connective tissue covered by a single layer of flattened epithelial cells called mesothelium. Its job is usually lubrication.
Let's get into the classification of epithelia themselves, starting with the single-layered ones, unilaminar or simple epithelia. Their shape tells a story, doesn't it?
It really does. The simplest shape is squamous. These are extremely flattened cells, like tiles or scales. We call them squames. Their cytoplasm can be incredibly thin, sometimes just 0.1 micrometers.
Wow. And why so thin?
Speed. Their primary function is rapid diffusion. Gases across lung alveoli, fluid filtration in kidney corpuscles. Minimum barrier thickness means maximum speed. And if you look at them from the surface, their edges often interlock in complex, wavy patterns. We call that tessellated. It adds mechanical stability to the thin sheet.
Okay, so flat means fast diffusion. What if the cell needs to be more metabolically active, like secreting or absorbing actively?
Then it needs more internal machinery, more mitochondria, more endoplasmic reticulum, and that requires more volume. So the cells become taller, cuboidal, or columnar.
Makes sense. Shape follows function.
Exactly. And these taller cells often have specialized surfaces. Think of the columnar cells lining the small intestine. They have a striated border, a dense carpet of thousands of regular finger-like projections called microvilli.
To increase surface area for absorption, right?
Massively increase it. Another specialization, even longer, are stereocilia. Despite the name, they're not true cilia. They're actually extremely long, sometimes branched microvilli. You find them in places like the epididymis, involved in absorption.
Now, what about that tricky one, pseudostratified? Looks layered, but isn't.
Ah, yes, the classic histology head-scratcher. It looks stratified because the cell nuclei are positioned at different heights within the layer. But the key defining feature and why it's simple, not stratified, is that every single cell rests on the basal lamina.
Even if they don't all reach the top surface.
Correct. Some might be shorter and not reach the lignin, but they all have a foothold on the foundation. The classic example is the ciliated columnar epithelium lining most of the respiratory tract.
With the cilia beating to move mucus.
Exactly. Those cilia beat in a coordinated way, creating the mucociliary rejection current, sometimes called the mucociliary escalator. It's a crucial defense mechanism, constantly sweeping mucus, trapped dust, and pathogens up and out of the airways.
Okay, so simple epithelia are often about transport or secretion. What about the multi-layered ones, multilaminar or stratified epithelia? They sound like they're built for toughness.
They absolutely are. You find them on surfaces subjected to significant mechanical stress or abrasion. Having multiple layers means that superficial cells can be worn away without compromising the barrier function.
And replacement comes from below.
Yes. There's continuous cell division in the basal layers nearest the connective tissue. As new cells are produced, they get pushed upwards, progressively flattening as they migrate, and are eventually shed from the surface. And these layers are held together incredibly strongly by numerous desmosomes.
The most common type is stratified squamous. And it comes in two main flavors, right? Keratinized and non-keratinized.
That's the key distinction. Keratinized stratified squamous epithelium is adapted for dry surfaces exposed to significant wear and tear, think epidermis, your skin, or the hard palate in your mouth.
And the keratinization means dead cells on top.
Exactly. The most superficial layer, called the stratum corneum, consists of flattened squames that are enucleate. They've lost their nucleus and are essentially dead bags packed full of tough keratin protein. Plus, there are lipids sealing the spaces between them. This creates a very tough, durable, water-resistant barrier.
Okay, and the non-keratinized version.
Non-keratinized stratified squamous epithelium is found on surfaces that also experience abrasion but are kept moist, protected from drying out. Examples are the lining of the esophagus, the mouth, the vagina. Here, the surface cells are still living and retain their nuclei right up until they are shed.
Is there an in-between state?
Sometimes, pathologically or due to chronic irritation, you might see perikeratinization, where the surface cells start to keratinize but retain remnants of their nuclei. This is distinct from the fully mature enucleate state of orthokeratinization seen in healthy skin.
Now, for a really specialized stratified type, urothelium, or transitional epithelium, found only in the urinary system.
That's right. Lining the ureters, bladder, and part of the urethra. It's uniquely adapted to withstand both the toxicity of urine and, crucially, extreme stretching.
Transitional, because it changes thickness.
Yes. When the bladder is empty and relaxed, the urothelium might be, say, 4 to 6 cells thick. But when the bladder fills and stretches, the epithelium thins out dramatically, maybe down to only 2 or 3 cells thick. The cells rearrange and flatten.
Let's talk about those surface cells, the umbrella, or dome cells. They're quite remarkable, aren't they? Especially dealing with urine.
They really are architectural wonders. They're very large and often binucleate, 2 nuclei, or even polyploid sometimes, containing up to 8 sets of chromosomes, making them octoploid.
8 sets. Why so much DNA?
The thinking is it provides genetic redundancy. It allows the cell to cope with the constant low-level DNA damage caused by toxic substances concentrated in urine, and maybe helps handle the sheer physical stress of stretching. They also have a very specialized apical membrane facing the urine.
What's special about it?
It's studded with rigid-looking areas called intramembranous plaques. These are protein assemblies that make the membrane unusually stiff. When the bladder is empty, excess membrane containing these plaques is folded inwards into the cytoplasm. When the bladder stretches, these folds smooth out, the plaques become part of the surface again, stiffening the membrane and helping prevent urine from leaking back into the tissues.
Amazing adaptation. Lastly, in epithelia, let's revisit those myoepithelial cells. Part epithelial, part muscle.
Kind of. They're often called basket cells because they form a network, like a basket, around the secretory units, acini, of many glands, salivary glands, mammary glands, sweat glands.
And they contract.
Yes. They contain actin and myosin filaments, the same proteins that make muscle contract. They're usually stimulated by nerves or hormones. When they contract, they squeeze the secretory unit.
Helping to push the secretion out into the duct.
Exactly. They provide the initial push to get the product flowing. But despite having contractile machinery like muscle, their origin is epithelial. And the proof is that they still express keratin intermediate filaments, the hallmark of epithelia, alongside their actin and myosin. A true hybrid.
All right, we've covered barriers and linings. Now let's move to the body's factories. Glands. Tissues specialized for making and releasing stuff, usually macromolecules.
That's their core function: synthesis and secretion. Often, their products modify the environment on a free surface, like digestive enzymes, or regulate processes elsewhere in the body, like hormones.
How do we first classify them? Based on where the product goes.
That's the primary division. Exocrine glands are defined by having a duct. They secrete their product through this duct onto a surface, either the body's exterior, like sweat glands on the skin, or onto an internal surface continuous with the exterior, like salivary glands secreting into the mouth, or pancreatic enzymes into the gut.
Okay, exocrine means ducts, the alternative.
Endocrine glands. These are ductless. They release their products, which we call hormones, directly into the surrounding interstitial fluid. From there, the hormones diffuse into nearby capillaries and enter the bloodstream to travel throughout the body. Think thyroid gland, adrenal glands, pituitary gland.
Got it. And there's a third, more local type.
Yes, paracrine cells. These cells secrete signaling molecules, but these molecules only act locally, diffusing short distances to affect adjacent target cells. Many of these are scattered neuroendocrine cells acting as local regulators.
Let's dive into the how of secretion. The different mechanisms cells use to release their products. This tells us a lot about cell biology.
It really does. The most common and, let's say, elegant method is merocrine secretion.
How does that work?
The cell packages its product, maybe proteins or mucus, into membrane-bound vesicles inside the cell. These vesicles then move to the cell surface. Their membrane fuses perfectly with the plasma membrane, and the contents are released outside. This whole process is called exocytosis.
And the cell itself remains intact.
Completely intact, no loss of cytoplasm. It's a very clean, controlled release. It involves specialized proteins on both the vesicle and the cell membrane to ensure the fusion happens correctly. Most glands, including pancreatic acinar cells and, by modern classification, sweat glands, use this merocrine mechanism.
Okay, that's the neat one. What's the next mode? Apocrine.
Apocrine secretion is a bit messier. Here, some of the cell's apical cytoplasm, the part near the lumen, gets pinched off along with the secretory product. The product itself might accumulate as droplets within the cytoplasm first.
So the cell loses a bit of itself.
Yes. The classic example, really the main undisputed one in humans, is the secretion of milk fat by mammary gland cells. The large lipid droplet bulges from the apical surface, taking a wrapping of cell membrane and a small amount of cytoplasm with it as it buds off.
Like sending the product off with a little packed lunch from the cell.
Haha, exactly. A good analogy.
And the most extreme method, holocrine.
Holocrine secretion is essentially cellular self-destruction for the cause. The entire secretory cell fills up with its product, and then the whole cell disintegrates, releasing its contents.
The cell dies in the process.
It does. Its remnants become part of the secretion. The prime example is sebaceous glands in the skin, which produce the oily sebum. New cells differentiate, fill with sebum, die, and rupture to release it into the hair follicle. It's a terminal differentiation process.
Okay, modes of release covered. What about gland structure and the type of product?
Structurally, glands range from the very simple, a single unicellular gland, like the goblet cell producing mucus in the gut lining, all the way to complex multicellular glands.
And the multicellular ones can have different shapes.
Yes, they can be simple tubes, simple tubular, coiled tubes, coiled tubular, or have flask-shaped secretory units called acini, simple acinar, or they can be highly complex with branching ducts, compound glands, leading to tubular or acinar secretory portions.
And functionally based on the product, mucus versus serous.
Right. We can often tell the product type just by looking at the cells histologically. Mucus glands secrete mucins, which are glycoproteins that become very hydrated to form mucus. Because the mucus gets washed out during standard tissue preparation, the cells often look pale and frothy, and their nuclei tend to be flattened against the basal side of the cell.
Okay, pale and frothy means mucus. What about serous?
Serous glands typically secrete watery fluids rich in proteins, often enzymes like digestive enzymes or glycoproteins. Their cells usually have round, centrally located nuclei, and their cytoplasm is often packed with visible, protein-rich secretory granules that stain pinkish with eosin, eosinophilic. The parotid salivary gland is a classic serous gland.
Can you have both types in one gland?
Absolutely. Mixed glands contain both mucus and serous secretory units. A common arrangement, seen in the submandibular salivary gland, is to have mucus acini capped by a little crescent of serous cells, called a serous demilumen. Both cell types empty into the same lumen.
Focusing back on endocrine cells for a moment, the hormone producers. Their location relative to blood vessels is key, right?
It's absolute critical. Since they secrete directly into the bloodstream, endocrine cells are always intimately associated with dense networks of capillaries, and often these capillaries have fenestrated endothelia.
Meaning the capillary walls have little pores.
Exactly. Tiny windows or pores in the endothelial cells that line the capillary. These fenestrations allow large molecules like protein or peptide hormones to pass easily and rapidly from the interstitial fluid into the blood circulation.
And some endocrine cells aren't clustered in distinct glands, are they? They're scattered.
That's the dispersed neuroendocrine system. You find individual endocrine cells scattered within the epithelium of other organs, particularly the digestive tract, like G cells producing gastrin, and the respiratory system. These cells typically make peptide hormones, store them in granules near their basal surface, close to the capillaries, and release them to act locally or enter the circulation. They often have prominent RER and Golgi, indicating high synthetic activity.
So how is all this secretion, exocrine and endocrine controlled? It must be tightly regulated.
It is. Regulation involves both the endocrine system itself, hormones controlling other hormones, and the autonomic nervous system. Feedback loops are fundamental.
Like negative feedback.
Negative feedback is the most common physiological control mechanism. A hormone is released, travels to its target, causes an effect, and often the hormone itself or the effect it produces signals back to the original gland to decrease further hormone production. Think of testosterone inhibiting its own release from the testes and pituitary. It keeps things in balance.
Is there positive feedback too?
Positive feedback is much rarer but crucial in specific situations. Here, the hormone or its effect signals back to amplify the original stimulus, creating a surge. The classic example is the surge of oosestradiol before ovulation, which positively feeds back on the adenohypophysis, anterior pituitary, causing a massive release of luteinizing hormone.
So, hormonal control via feedback. What about direct nerve control?
Yes, many glands receive direct input from autonomic secretomotor nerve fibers. These nerves synapse directly on the secretory cells or surrounding cells, like myoepithelial cells, to stimulate or inhibit secretion. Think of salivating when you smell food that's neural controlled.
And local signals.
Right, paracrine signals play a role too. For instance, histamine released by mast cells can locally affect blood vessels and potentially glandular secretion in the vicinity. And we can't forget the mechanical help from myoepithelial cells in exocrine glands. Their contraction, often triggered by nerves or hormones, physically squeezes the product out.
Okay, let's zoom in on that critical boundary layer we touched on earlier, the structure separating epithelia from the underlying connective tissue. There's some specific terminology here that's important for real mastery, isn't there?
Right.
Basement membrane versus basal lamina.
Absolutely crucial distinction. Under a standard light microscope, you often see a distinct line or layer beneath the epithelium that stains well with stains for carbohydrates, like PAS stain. This visible layer is traditionally called the basement membrane.
But that's the low-resolution view.
Exactly. When you use an electron microscope, which has much higher resolution, you can see that this basement membrane actually consists of finer structures. The layer immediately adjacent to the basal surface of the epithelial cells, the one they actually produce, is the basal lamina. It's a delicate, felt-like network, typically around 80 nanometers thick.
So the basal lamina is part of the basement membrane, the part made by the epithelium.
Correct. And underneath the basal lamina, there's usually another layer of matrix material, the reticular lamina. This contains different types of fibrils, like type 3 collagen, and is produced by the connective tissue cells, fibroblasts, below. It essentially merges with the rest of the connective tissue. So basement membrane, light microscopy term, basal lamina plus reticular lamina, electron microscopy components.
What about the lamina lucida? I remember seeing that in older diagrams, a clear space between the cell and the basal lamina.
Ugh, the lamina lucida. For a long time, it was shown in diagrams as an electron-lucent, clear gap. However, with improved tissue preparation techniques like rapid freezing instead of chemical fixation, this lucent zone often disappears or is much reduced. So the current thinking is that the lamina lucida might largely be an artifact of older preparation methods. The key functional components are the basal lamina itself and its connection to the cell above and the reticular lamina below.
Okay, focusing on the basal lamina then, the epithelial product, what's it actually made of? Its molecular architecture. You called it a layer cake before.
Let's stick with that analogy. It's a highly organized meshwork of specialized glycoproteins and proteoglycans. The foundational layer, maybe the sponge cake layer, is built primarily from laminin.
Laminin, what does it do?
Laminin is a large, flexible, cross-shaped glycoprotein. Crucially, laminin molecules have the ability to self-assemble into a two-dimensional sheet-like network. It forms the initial scaffold of the basal lamina. In fact, very early embryonic basal laminae are almost entirely laminin.
So laminin forms the sheet. What gives it structural reinforcement, like the rebar in concrete?
That would be type 5e collagen. Unlike the thick type I collagen sorbroles found in tendons, type 5e collagen molecules assemble into a complex, mesh-like sheet that integrates with the laminin network, providing tensile strength and stability to the lamina.
Okay, laminin sheet, type 5 collagen mesh.
Okay,
How are they linked together? What's the molecular glue or cross-linker?
good question. Two key players here. Nitogen, also called intactin, is a rod-like molecule that acts as a bridge, cross-linking the laminin network to the type 5 collagen meshwork. And then there's perlecan, which is a very large proteoglycan containing heparin sulfate side chains. Perlecan helps organize the structure, binds to many other components, and contributes to the filtration properties of the basal lamina due to its negative charges.
Understanding this recipe matters because variations cause specific diseases, right?
Right.
You mentioned kidney problems.
Exactly. The precise molecular composition of the basal lamina can vary slightly between different tissues, and this has major clinical implications. Alport syndrome, for example, is a genetic disease causing kidney failure. It results from mutations in the genes encoding specific chains of type 4 collagen that are predominantly expressed in the glomerular basal lamina of the kidney. The filter breaks down.
So a defect in type 4 collagen hits the kidney filter hard, but might not affect, say, skin basement membrane as much.
Precisely because the exact isoforms or composition might differ or the functional demands are different. Another example is Goodpasture syndrome, an autoimmune disease where the body mistakenly produces antibodies that attack a specific component of the type 4 collagen in the basal laminae of the kidney glomeruli and lung alveoli. Again, devastating consequences for those organs.
Are there other specialized basal lamina components with unique roles?
Oh yes, different types of laminin exist. Laminin 2, for instance, is crucial for the basal lamina surrounding muscle cells and Schwann cells, which insulate peripheral nerves. And think about the neuromuscular junction, the communication point between nerve and muscle. The basal lamina there contains a special proteoglycan called agrin.
Agrin. What's its job?
Agrin, secreted by the nerve terminal, gets incorporated into the basal lamina and plays a critical role in clustering acetylcholine receptors on the muscle cell membrane directly opposite the nerve ending. It essentially tells the muscle cell exactly where to listen for the nerve signal. It's an instructive molecule embedded in the matrix.
So summing up, this basal lamina isn't just passive support, it's doing a lot.
A huge amount. Its key functions include
Acting as selective permeable barriers. The glomerular basal lamina in the kidney is the prime example, filtering blood based on size and charge. The negative charges from proteoglycans like perlecan repel negatively charged proteins, providing anchoring and stability. It anchors the epithelium firmly to the underlying connective tissue. Structures called hemides mucosomes on the basal surface of epithelial cells, like skin keratinocytes, physically link the cell's internal cytoskeleton through the basal lamina into the connective tissue,
3.
exerting instructive effects on the cells above it. The basal lamina influences cell polarity, which weighs up, differentiation, survival, and even the rate of cell division. Cells need that connection for proper behavior,
And finally,
serving as pathways for migration. During development and regeneration, the basal lamina can provide tracks that guide migrating cells, like nerve axons growing out to reinnervate a muscle after injury. It helps ensure connections are reestablished correctly. And pathologically, thickening and disorganization of the basal lamina are hallmarks of conditions like diabetes, especially affecting kidney and blood vessel BMs, and chronic inflammation like glomerulonephritis.
all right, we've mastered epithelia and their foundation. Now we cross the basal lamina into the realm where the matrix is king, connective tissues. Here, the extracellular matrix, ECM, dominates, right?
Completely. Connective tissues are defined by having abundant intercellular material, the ECM composed of fibers and amorphous ground substance, with the cells often being relatively sparse.
And what are the big picture roles of these tissues?
They're incredibly versatile. Primarily structural, they provide the mechanical framework, strength, and form for organs and the body as a whole. They're also crucial for defense, as they are the main battleground for the immune system, hosting many resident and migrant immune cells. And they have trophic and morphogenetic roles, influencing cell growth, differentiation, and tissue repair through the molecules within the ECM and signals from the cells.
Let's start with the cells that live there permanently, the resident cells, the builders and maintainers.
Top of the list are the fibroblasts. They are the most common cell type in general connective tissue. Their job is to synthesize and secrete almost all the components of the ECM. The collagen fibers, the elastin fibers, the ground substance molecules.
Do they always look the same?
No, they exist on a spectrum of activity. An active fibroblast, busy-making matrix, typically has a larger, more open nucleus, euchromatic, and lots of rough endoplasmic reticulum, RER, and Golgi apparatus. When less active, they become smaller, more spindle-shaped, with a darker nucleus, and we sometimes call this inactive state a fibrocyte.
You mentioned fibroblasts making collagen. What about those fine branching fibers, reticular fibers?
Fibroblasts make those too. Reticular fibers are primarily composed of type 3 collagen. Fibroblasts synthesize this type 3 collagen to create delicate branching networks, reticular tissue that form a supportive meshwork within soft organs like the liver, spleen, and lymph nodes.
What happens when there's an injury? Do fibroblasts change?
They can undergo a fascinating transformation into myofibroblasts. These cells ramp up their matrix production but also gain contractile properties, similar to smooth muscle cells. They express alpha-smooth muscle actin in stress fibers.
And why is that useful?
They're absolutely crucial for wound contraction. During wound healing, myofibroblasts align themselves and pull the edges of the wound together, physically shrinking the defect. It's a vital part of the repair process. Their origin is complex too. They can rise from resident fibroblasts, from circulating precursor cells, or even from other cell types like parasites around blood vessels, showing remarkable cellular plasticity.
But this repair mechanism can go wrong, can't it? Leading to scarring or fibrosis.
Exactly. Fibrosis is essentially wound healing gone wild. In diseases like liver cirrhosis or idiopathic pulmonary fibrosis, there's excessive deposition of stiff, scar-like ECM. A key problem in pathological fibrosis is that the repair process doesn't switch off properly. Normally, after a wound is closed, myofibroblasts and inflammatory cells should undergo programmed cell death apoptosis. In fibrotic diseases, they often fail to die, leading to persistent matrix production and tissue hardening.
Is this the same as scar formation in the brain?
No, scar formation in the central nervous system is quite different. Glial scars are formed primarily by astrocytes, a type of glial cell, not by fibroblasts, which are generally excluded from the CNS parenchyma. It's a distinct process with different cellular players.
Okay, moving from the matrix builders to the energy specialists. Adipocytes or fat cells? Two main types, you said.
Yes, dramatically different in structure and function. The most common is white fat, composed of unilocular adipocytes. Unilocular means they contain one single large lipid droplet, mostly triglycerides, that fills almost the entire cell, pushing the nucleus and cytoplasm to a thin rim at the periphery. Their main job is energy storage.
And the other type, brown fat.
Brown fat is made of multilocular adipocytes. These cells contain numerous small lipid droplets scattered throughout the cytoplasm, and critically, they are packed with mitochondria. These mitochondria have unusually large, numerous cristae.
Why so many mitochondria?
Because brown fat's primary function is not energy storage, but heat generation, thermogenesis. The mitochondria in brown fat uniquely express a protein called UCP1, or uncoupling protein 1. UCP1 short-circuits the normal process of ATP production. Instead of using the energy from breaking down fats to make ATP, it allows the energy to be released directly as heat. It's like running the cell's furnace on high.
And fat tissue isn't just passive storage anymore, is it? We know it acts like an organ itself.
Absolutely. Adipose tissue is now recognized as a major endocrine organ. It secretes a whole host of signaling molecules called adipokines, the most famous being leptin, which signals satiety to the brain and regulates energy balance. Others influence insulin sensitivity, inflammation, and more. And an important point about obesity. In adults, weight gain is primarily due to existing white fat cells getting larger, hypertrophy, not usually a massive increase in the number of fat cells, hyperplasia, although that can happen too, especially in severe obesity starting in childhood.
We should also mention the reserve troops, the mesenchymal progenitor cells.
Yes, these are crucial, but often hard to spot histologically. They are relatively undifferentiated pluripotent cells derived from the embryonic mesenchyme that persist in small numbers within adult connective tissues. They retain the ability to divide and differentiate into various connective tissue cell types: fibroblasts, adipocytes, chondrocytes, cartilage cells, osteoblasts, bone cells. They are vital for lifelong tissue turnover, repair, and regeneration, and hold immense therapeutic promise.
Okay, those are the residents. Now for the cells that move in and out, the defense force, the migrant cells. Let's paint a picture. You get a splinter, breaks the skin, bacteria get into the connective tissue underneath. Who responds first?
The first responders already on the scene are the resident mast cells. They are strategically located, often near blood vessels. They are packed with large granules containing potent inflammatory mediators like heparin, an anticoagulant. Histamine increases vascular permeability and causes vasodilation, and enzymes like tryptase.
And when the splinter hits, they release this stuff.
Yes. Physical trauma or antigens can trigger them to degranulate, rapidly releasing these mediators. This causes the classic signs of inflammation: redness and heat from vasodilation, swelling from leaky capillaries, which helps bring plasma proteins and more immune cells to the site. Mast cells essentially sound the alarm and call for backup. They look a bit like basophils from the blood, but they are a distinct lineage developing in the tissues.
Okay, alarms out, a reinforcement's called. Who are the first professional cleaners and fighters to arrive from the bloodstream?
Key players arriving quickly are neutrophils. These are a type of granulocyte, highly phagocytic, especially towards bacteria. They engulf and destroy the invaders. Also arriving are macrophages. These large phagocytes, 15 to 20 mm, actually differentiate from monocytes that migrate out of the bloodstream. Their job is massive cleanup, engulfing bacteria, dead cells, debris, and the splinter itself. They're part of the widespread mononuclear phagocyte system, which includes specialized macrophages in different tissues, like Kupffer cells in the liver or microglial cells in the brain.
What about the more specialized immune cells, the lymphocytes?
They arrive too, playing roles in coordinating the response and providing specific immunity. B lymphocytes, when activated by antigens, like bacterial components, proliferate and differentiate into plasma cells.
Ah, the antibody factories.
Exactly. Plasma cells are dedicated to synthesizing and secreting vast amounts of antibodies, immunoglobulins, specific to the invading pathogen. They have a very characteristic appearance: extensive RER filling their cytoplasm, a prominent Golgi apparatus, and an eccentric nucleus with clumped chromatin, often described as a clock face pattern. A key point: mature plasma cells are terminally differentiated. They focus solely on antibody production and do not divide.
And the T lymphocytes.
T lymphocytes have diverse roles. Some cytotoxic T cells directly kill infected host cells, like virus-infected cells, or foreign cells, like in graft rejection. Others, helper T cells, are crucial coordinators, interacting with B cells and macrophages to orchestrate the overall immune response. They mature in the thymus, hence the T. We might also see eosinophils, another type of granulocyte, whose numbers increase particularly in parasitic infections or allergic reactions.
So connective tissue is a dynamic place, full of resident builders and migrant defenders interacting within the matrix. Let's look closer at that matrix itself, starting with the fibers that give it strength, the fibrous matrix, dominated by collagen.
Absolutely dominated. Collagens are the most abundant proteins in the entire body, making up about 30% of our total protein mass. They are a huge family, with at least 29 different types known currently. All share a characteristic triple helix structure, formed by 3 polypeptide chains wrapping around each other, and they are rich in specific amino acids like glycine, proline, and modified forms like hydroxyproline.
Let's compare the main types to get a feel for their different jobs. Type I.
Type I collagen is the main structural workhorse. It's the most abundant type, forming thick, strong fibrils found in tissues that need to resist high tensile forces. Think tendons, ligaments, bone, the dermis of the skin. Under the electron microscope, these fibrils show a very distinct cross-banding pattern repeating every 65 or 67 nanometers, which reflects the regular packing of the collagen molecules. The thickness of these type I fibrils often correlates with the tensile load the tissue normally bears. Defects in type I collagen structure or synthesis cause conditions like Ehlers-Danlos syndrome, some types, and osteogenesis imperfecta, brittle bone disease.
Okay, type UN is the thick rope. What about type 3, the reticular fibers you mentioned?
Type 3 collagen forms much finer fibrils that don't bundle together as thickly as type 1. Instead, they form delicate branching networks, the reticular fibers. These provide a supportive mesh or scaffold within expandable organs like the liver, spleen, lymph nodes, and bone marrow. They're also abundant in wound healing and early developmental tissues. Because they have more carbohydrate groups attached, they don't stain well with standard H&E, but can be specifically stained black with silver salts. Argyrophilic.
Are there collagen types that form sheets instead of fibers?
Yes, we already met type 4 collagen, which forms meshworks in basal laminae. Other types, like type 8 and type X, also form networks rather than fibrils, contributing to specialized structures.
So collagen provides tensile strength, resists pulling. What provides the stretch and recoil?
That's the job of elastin. Elastin is a protein that allows tissues to stretch considerably and then snap back to their original shape, like a rubber band. It's rich in hydrophobic amino acids, like glycine, valine, and proline, and is highly cross-linked by unique covalent bonds involving lysine residues, forming structures called desmosine and isodsmosine. These cross-links are essential for elasticity. Elastin can form fibers, often interwoven with collagen, or arranged into sheets like the elastic lamellae in the walls of large arteries, like the aorta.
And problems with elastic fibers lead to?
Conditions like Marfan syndrome. This genetic disorder is caused by mutations in the gene for fibrillin-1, a glycoprotein essential for organizing elastin into functional fibers. Patients often have weakened blood vessel walls, risk of aortic aneurysm, skeletal abnormalities, and eye problems, all related to defective elastic tissue.
Okay, we have the fibers collagen for strength, elastin for recoil. What about the stuff filling the spaces between them, the interfibrillar matrix or ground substance?
This is the hydrated, gel-like material that fills the space. It's composed primarily of glycosaminoglycans, JGs, proteoglycans, and adhesive glycoproteins.
Let's break that down. GAGs.
Glycosaminoglycans, or GAGs, are long, unbranched polysaccharide chains, typically composed of repeating disaccharide units. Crucially, they are highly polyanionic. They carry a large number of negative charges due to sulfate and carboxyl groups.
Why are those negative charges important?
They attract positively charged ions like sodium, Na+, which in turn osmotically attract huge amounts of water. This creates a highly hydrated, swollen gel that resists compressive forces very effectively. Think of cartilage resisting compression in your joints. That's largely due to GAGs. Most GAGs, like chondroitin sulfate, keratin sulfate, heparin sulfate, are covalently linked to a core protein to form proteoglycans.
Is hyaluronic acid different?
Yes, hyaluronan is unique among GAGs. Firstly, it's enormous. A single molecule can be incredibly long. Secondly, it's not sulfated, and it's not typically covalently linked to a core protein to form a proteoglycan. Instead, many proteoglycans can attach non-covalently to a central hyaluronan molecule, forming massive structures called proteoglycan aggregates. These aggregates are incredibly effective at trapping water and resisting compression, particularly in cartilage. Hyaluronan also contributes to the viscosity of synovial fluid, lubricating joints.
And the third component of ground substance, adhesive glycoproteins.
These are large molecules with multiple binding domains that act like molecular glue, linking cells to the matrix components and linking different matrix components together. They also play key roles in cell signaling, migration, and differentiation.
Key example.
Fibronectin is a major one. It's a dimer linked by disulfide bonds with binding sites for collagen, heparin, ajongi, and, importantly, cell surface receptors called integrins. By linking cells to the matrix via integrins, fibronectin mediates cell adhesion and migration. Different forms, isoforms of fibronectin, generated by alternative splicing of its mRNA, are important in embryonic development and wound healing. We already discussed laminin, which is the primary adhesive glycoprotein in basal laminae, forming meshworks and binding to integrins. Another important one is tenascin, a large glycoprotein composed of 6 subunits joined together. It appears particularly during embryonic development and wound repair, where it's thought to modulate cell adhesion and guide cell migration, including neuronal axon growth.
So, putting it all together, cells, fibers, ground substance, how do we classify the different types of connective tissue based on their organization?
We generally classify them based on the density and arrangement of the fibers. Irregular connective tissues have fibers oriented in multiple directions, resisting stresses from various angles. Loose irregular CT, also called areolar tissue, has relatively sparse fibers and abundant ground substance in cells. It's the most widespread type, found beneath epithelia, as lamina propria, around blood vessels and nerves, filling spaces. It binds structures but allows considerable movement. Dense irregular CT has much thicker bundles of collagen fibers running in various directions, with less ground substance and fewer cells. It provides significant strength and resistance to tearing. You find it in the dermis of the skin, capsules surrounding organs, and sheaths around nerves.
Okay, that's irregular. What about regular?
Regular connective tissues have fibers arranged in a highly ordered parallel fashion, adapted to resist strong tensile forces in one specific direction. Dense regular CT is the classic example, forming tendons, connecting muscle to bone, and ligaments, connecting bone to bone. Here, the collagen fibers are tightly packed in parallel arrays, and the fibroblasts, tenocytes and tendons, are compressed into rows between the fibers. Fasciae, sheets of CT, can also be dense regular. There's also elastic tissue, a type of dense regular CT where elastic fibers predominate, found in structures like the ligamenta flava of the vertebral column and the walls of large arteries.
And that embryonic type,
Yeah.
mucoid tissue.
All right, mucoid tissue is a very loose, gel-like connective tissue found primarily in the embryo, particularly in the umbilical cord, where it's called Wharton's jelly. It consists mainly of abundant, highly hydrated ground substance, rich in hyaluronan, with scattered mesenchymal cells and fine collagen fibers. It mostly disappears after birth, but persists in a few places like the vitreous body of the eye and the nucleus pulposus of intervertebral discs in adults.
We've seen incredible specialization, but cells can sometimes change their identity, can't they? This idea of metaplasia.
Yes, metaplasia represents a switch from one fully differentiated cell type to another fully differentiated cell type. It's usually an adaptive response of a tissue to chronic irritation or a changed environment.
So the cells themselves transform.
Not quite. It's generally thought that the existing differentiated cells don't change. Instead, the tissue stem cells or progenitor cells, which normally replace lost cells, alter their pathway of differentiation in response to the persistent abnormal stimulus. They start producing a different, often more resistant cell type. Postnatally, metaplasia is almost always considered pathological, even if adaptive initially.
Can you give us the classic examples where this happens?
The very common one is squamous metaplasia in the airways of smokers. The normal lining is pseudostratified ciliated columnar epithelium, specialized for mucus secretion and clearance. Chronic irritation from cigarette smoke causes the stem cells to switch to producing stratified squamous epithelium instead.
Which is tougher, right? More resistant to the smoke.
It is tougher, yes, better at resisting physical and chemical injury. But the trade-off is you lose the protective functions of the original epithelium. No mucus secretion, no cilia to clear debris. This makes the lungs more prone to infection and is considered a pre-malignant change, increasing the risk of developing squamous cell carcinoma.
And the other way around, columnar replacing squamous.
That happens in Barrett's esophagus. Here, chronic acid reflux from the stomach damages the normal stratified squamous lining of the lower esophagus. In response, the stem cells can switch to producing a columnar, mucus-secreting epithelium, similar to that found in the stomach or intestine, gastric or intestinal metaplasia. Again, while this new lining might be more resistant to acid, it carries a significantly increased risk of developing adenocarcinoma of the esophagus. The key danger in metaplasia is this increased susceptibility to malignant transformation.
Okay, a final look at some specific composite structures. We mentioned mucosa, mucous membrane earlier. Let's recap its structure.
Right. Mucosa lines cavities that open to the exterior digestive, respiratory, urinary, reproductive tracts. It typically consists of 3 layers. 1, the epithelial lining, which varies depending on the location. There's a simple columnar in the stomach, stratified squamous in the esophagus. 2, an underlying layer of loose connective tissue called the lamina propria, which supports the epithelium, contains blood vessels, nerves, and immune cells. 3, often, but not always, a thin layer of smooth muscle called the muscularis mucosae at the base, which allows the mucosa some independent movement.
And the defining feature is often mucus production. What exactly is mucus, chemically and physically?
Mucus is a viscous, slimy secretion primarily composed of water and large glycoproteins called mucins. Mucins are the key component. They have a protein core with numerous long, complex carbohydrate chains attached, oligosaccharides.
And these carbohydrate chains are important.
Hugely important. They are highly branched and carry negative charges, causing them to attract and trap large amounts of water via hydrogen bonding. This hydration is what gives mucus its characteristic gel-like, viscous, and slippery properties. Mucin synthesis is a major cellular effort involving protein synthesis in the RER, extensive glycosylation, adding the sugars, in the Golgi apparatus, and packaging into large secretory granules, mucinogen granules, before release via exocytosis, migrant secretion.
Functions: lubrication, protection.
Both and more. It lubricates surfaces, allowing food to pass easily down the esophagus or protecting airways from drying. It acts as a physical barrier, trapping inhaled particles or microorganisms. It binds water, preventing dehydration of underlying cells, and it contains defensive molecules like antibodies and enzymes.
And failure to properly hydrate mucus has devastating consequences, like in cystic fibrosis.
Exactly. Cystic fibrosis, CF, is a genetic disease caused by mutations in the CFTR gene, which codes for a chloride ion channel protein found in epithelial cells. When this channel doesn't function properly, chloride transport across the epithelium is impaired. Since water movement by osmosis follows ion movement, especially chloride and sodium, water secretion is reduced. This results in abnormally thick, sticky, dehydrated mucus in various organs, particularly the lungs, leading to obstruction and chronic infections, pancreas, blocking enzyme secretion, and intestines. It tragically highlights the importance of proper mucus hydration.
Finally, the other major membrane lining internal cavities, serosa, serous membrane.
Serosa lines the closed body cavities, the pleural cavity around lungs, pericardial cavity around heart, and peritoneal cavity in the abdomen. It consists of a single layer of flattened squamous epithelial cells called mesothelium, supported by a thin layer of underlying loose connective tissue containing blood vessels and lymphatics.
Mesothelium, that's epithelial.
Yes, despite its simple squamous appearance, mesothelium is derived from embryonic mesoderm, like connective tissue, but expresses keratin filaments, confirming its epithelial nature. Its main function is to produce a thin, watery serous fluid that lubricates the surfaces of the cavity, allowing organs within the cavity, like the heart beating or the intestines moving, to glide smoothly against each other and the body wall with minimal friction. #NagOutro. So we've journeyed from the basic 4 pillars all the way down to the molecular nuts and bolts, the specific collagens, laminins, JAGs, junctions that build these tissues. It's been quite a deep dive.
It really has, and I think the biggest takeaway for me is just how active and instructive the extracellular matrix really is. It's not just packing material, it's constantly signaling, guiding, and supporting the cells.
Absolutely. And understanding that intricate interplay is key. Think about Alport syndrome, a devastating kidney disease caused by a defect in just one specific type of collagen in the basal lamina. Or how fibrosis results from myofibroblasts essentially forgetting to switch off their matrix production. Structure, function, and pathology are completely intertwined at this level.
Which makes this knowledge immediately relevant, doesn't it? Understanding disease isn't just about the symptoms, it's about understanding these fundamental failures in tissue architecture and cellular communication.
Precisely. And that understanding opens up possibilities.
Which brings us to that final thought. We saw how metaplasia demonstrates the body's inherent, albeit sometimes dangerous, cellular plasticity, the ability of stem cells to change their fate based on signals.
Right. Cells can change track.
So the provocative question is, if we truly master the molecular signals within the ECM and understand the potential of those mesenchymal progenitor cells we talked about, could we learn to guide that plasticity? Could we potentially poke cells down specific pathways to actively repair damaged tissues or even reverse fibrotic diseases, moving beyond just managing symptoms?
That's the frontier, isn't it? Harnessing that innate plasticity, informed by this deep knowledge of tissue architecture, to develop truly regenerative therapies. It's a compelling future to think about.
Indeed it is. A lot to ponder. Thank you for joining us on this deep dive into the foundations of tissue architecture. We'll see you next time.